Hello, can everyone hear me? Okay. So good morning. It is my great pleasure to introduce today's Grand Round speaker, Dr. Patricia Musolino, my personal mentor and hero. She is a distinguished critical care and vascular neurologist, renowned for her expertise in gene therapy for white matter and cerebral vascular disorders. She leads both the MGB gene therapy and genetic vasculopathies team and the pediatric stroke and cerebral vascular program here at MGH. Her clinical and research work is dedicated to translating discoveries in human genetics and into practical clinical applications for these disorders. Through her collaborative efforts with clinicians, scientists, patients, and advocacy groups, she has established an international network of resources aimed at accelerating the development of targeted therapies and improving the lives of children and young adults affected by vascular diseases. Her groundbreaking work has earned her a number of prestigious awards, including recognition from the NINDS, the Hearst Foundations, the Child Neurology Foundation, and the MGH Executive Committee on Research. In 2017, she also received the Herbert Pardes Clinical Research Excellence Award from the National Clinical Research Forum for her pioneering work on the first gene therapy trial for a cerebral demyelinating disorder in children. We are delighted to have her here today to share her insights into genetic therapies for severe neurovascular disorders in children. Please join me today in welcoming Dr. Musolino.
Thank you, Carrie. Thank you. And thank you, everybody on the Zoom and here in the room. It's always a privilege to be here. For those of you that don't know it, I come from very far away from the Ether Dome, like 7,000 miles south. So every time that I embody this place and I walk through the Boomfish building, and if you guys haven't done it, do it. It starts to explain why we can do the things that we can. We are stepping on giants. We had the best mentorship. We had the best minds, mentees, students, residents. So it's always warming my heart to be here. And I look really forward to the questions and telling you more about what we've been trying to do and tackling over the recent years. These are my disclosures. I have a scientific research contract with Angea Biotherapeutics, Minorix. Otherwise, I'm consultant, but most of our funding comes from NIH at the moment, and it's a larger collaboration group that I will introduce you to as well. What you're going to see would not have been possible without all the preliminary work that only philanthropy, the department funds, ECOR, and foundations made possible. So never, ever underestimate working with the real people closest to the clinic, either the patients, the foundations, and of course, your department who wants you to dive into new things. So don't try to do what gets funded. Do what really is in your heart, and that will take you far, far away. I do really own what I'm going to be presenting to a much larger group. There's more than 60 scientists between PhD to PI levels working on the work that I'm going to be presenting briefly. And you can see the expertise that was brought together. I'm the neurologist, pediatric neurologist that has been developing and following the pediatric stroke and cerebrovascular genetics for several years since graduation from my neurocritical care fellowship. And I lack the expertise on vascular, smooth muscle, endothelial biology that is way further out in the cardiovascular field. And that's why I work with Raj and Mark. We cannot develop technologies without the people that develop the technologies. It's not the same thing to use something because I have an idea than to develop it to meet that idea. And that's been on the enzyme side for the CRISPR-based editing and all the different genome editing. Natalie trying to get us nanoparticles that can get us to the vessels, which are our problem. And Casey Maguire doing the AAVs, which are currently moving towards the clinic. And the expertise in the disease, and you see David Chang, neurocritical care, attending, an expert in subarachnoid hemorrhage and neurovascular physiology and imaging that is critical for seeing our proof of concept realizing. And Shira and Carlos at NIH, Shira at Hopkins, with the expertise in one of the diseases that we are going to be mentioning today. I'm not going to go deeply into it. It's mostly Raj-led work, but that's another fatal cerebrovascular cardiovascular disease of childhood, genetic in kind. And you'll see that from mouse to human, brain to brain, there is a big arc to be met. And a lot of our translational work is only possible because Heather Gray-Edwards at UMass has taken us on as another translational program with the large animal models needed for biodistribution and toxicity and all the things that we need, even mechanistically speaking, to understand if we're going to try to do this. This is a little bit of how we are thinking about ourselves and what this group and collaboration has been working on. In the center, we have genome editors and all kinds of modulation of genes. We're also working on siRNAs. We're working on gene addition, which is the most traditional gene therapy you will be familiar with. There is a delivery platform to date. There is no human proven tool to deliver things to the vessels in a systemic way. You can put a stent, we can put a polymer with a stent embedded, but we don't have drugs or delivery platforms that can take you materials in a high efficiency to the endothelium and smooth muscle cells and the pericytes in the brain. So that's a big area to tackle, and to really do the translation, we need human data, and that's why we're trying to move with the most devastating disorders to the clinic. We do leverage the expertise, whomever has it. I don't need to be the expert on that mouse model, on that biology, on that mechanistic. I want to work with them. So if you can learn that early on, I'm betting your mentors are that kind of people. So Ben in the center, Casey and Natalie on the platform. Heather has enabled another loop in the preclinical, which is this bioreactor. We're able to test arteries from different animals and decide if the platform is translating to human arteries, even from samples coming from the OR. But in the meantime, it allows us to pick, by comparison to human, what large species we need to be doing our preclinical toxicology and biodistribution studies. So this has gotten a lot of traction. It's only possible because we have this collaboration, and people drive to UMass a couple times a week to get samples from Heather's lab, pig, cow, sheep, and they come to our bioreactor. It's a lot of fun. I gotta say, all of this is a lot of fun. We definitely have to come from the disease perspective that I think is the main core of why you're doing what you're doing. If you're a clinician scientist, why you're doing what you're doing. If you're a scientist, please do collaborate and team up with a clinician so you can, from the get-go, know where the barriers are. What are the unmet needs? What are going to be the biggest challenges when we are going to try to treat these kids? There is an expertise that comes from biotech and pharma. It's not mine. It's provided by people that have done it for decades. And that's thanks to the innovation office here at MGB is possible. And of course, a bunch of different collaborators. And Roger and Nathan, have only Roger in picture, have been leading the GZTI, and they really have empowered us with the CMC, the manufacturing side, in really advancing the progress on this. Again, this is work of the VILA. You can see at the Center for Rare Neurological Disorders, that Florian Eichler, my mentor, and probably why I do crazy things, I can blame it on him. He was the first one to take gene therapy to cerebral diseases in a large-scale trial. I was a resident in neurology, NM1, when he said, Patty, do you want to join this meeting with Bluebird Bio? And from there on, he hasn't stopped. And he tries to bring a therapy for every disease and every patient that he can, not by himself, but by building the team that requires it. Remember that your lab may be small, you're not alone. And this leads to our mission. We really want to develop gene therapies or genetic therapies, whatever you want to call them. This is not gene addition. This is just manipulating genetics to be able to modify biology, to do like transforming drugs that can really improve the quality of life of our patients, one patient at a time. And you're going to see some of these diseases, but what? You only have 55 patients in the world? Why are you want to put all these efforts in developing a therapy for them? Yes, there are places where we can de-risk because the unmet need is larger and immediate and urgent, and the alternative is death or devastating neurological disorder. And there are other places like diabetes and cardiovascular disease that we cannot dare to take these medications because the risk benefit is not yet proven to be safe even. So That's why one with all in mind, what we put and be able to do will de-risk the ability to expand to further indications in the rare and in the non-rare diseases. So if you haven't been thinking about cardiovascular, cerebrovascular disease, is still to this day, the #1 cause of death and disability. The #1 cause of neurological disability is cerebrovascular disease in humans, in the entire world, not just the developed world. Something we're not doing. If this is still the case, we cannot claim that we have phenomenal therapies. We still have a large unmet need. And I put on this side of the slide, on the right-hand side, what are the therapies that we have currently? And yes, ideally, everybody is educated, does a regular diet, sleeps, physical activity, and all the things that no one in this room probably is doing. So welcome to our clinics in the future. So, sorry to be so in disclosure, but it's the reality. And then we have all kinds of drugs to treat blood pressure, and still patients are 50% of the time, you can follow Brain Health Institute, and what this really means, they're not controlled in their blood pressure. There's still damage being caused. There's biology at play. Same thing with the lipid lowering medications, the statins. The adherence to them is not great. The access to care is not great. And you have to be in a lifespan of treatment to really benefit from them, meaning decades of taking the medication consistently and early enough before disease has been established. And then when everything has failed and you're getting symptomatic stroke, MI, we go and try to open the vessel. That's, to me, the last resort, medieval type of solution. I love it. That's what I did critical care. Don't get me wrong. It's the thrill of being able to make a difference for one person, but it's far from addressing the problem that we currently have. Still maybe with the way that we go in to try to de-risk some of these genetic therapies. So when you think about cardiovascular disease, you think about lipids and blood pressure. Well, 90% of cardiovascular and cerebrovascular disease is arterial, is the vessel that is diseased. That's what causes the symptoms. Whether there is metabolic, inflammatory, and other genetic conditions collaborating to produce that milieu, at the end of the day, it's a vessel that has been diseased. The wall is ruptured. The wall is narrowing because it's growing towards the inside with the plaque. And as you know, decades to develop a plaque. You start probably at 8 years of age in your aorta with atherosclerosis. So by now you already have it. And if you are a kid with a genetic condition, all of that can happen in 6 months of life. So that's where the biology being the same, convergent between the non-genetic and the genetic, can really be leveraged to de-risk some of the progress that we want to do. So I put this on the non-genetic side. We need effective treatments to heal vessels. If you want to think about what we're trying to do here, we want long-lasting systemic effects. We don't want to treat one occlusion of the coronary artery when the disease is everywhere in the body, and it's also affecting your small vessel in the brain, and you're developing vascular dementia. Like, you can look at these numbers. If you have coronary artery disease, you also have probably 60% chance of having already mild cognitive impairment from small vessel disease. So they are not in isolation. This is a system. It goes and is continuous. Your endothelium doesn't stop between your brain, your kidney, your liver, and your heart. It's a continuum. And then we want to alleviate the risk and the side effects. A lot of these medications also have side effects. A lot of the procedures have severe side effects. And the healthcare burden, of course, as you could imagine, is quite significant. In the rare arena, we know the gene, we know the root problem. We want to restore that pathway. And those pathways may be directly from the gene or a couple steps down from the gene. And that's where the biology then becomes convergent with the non-genetic disorders. And I'll show you some of that. We really want to go at specific mutations where we can have a much higher rate of success. Clinical trials in monogenic disorders are 7-fold more successful than clinical trials in non-genetic disorders. So you're more likely to get a successful clinical trial if you're treating a genetic disorder, because you're treating the root cause, even with all the risks that genetic therapies have. And then, of course, we want to prevent the symptoms. In this case, myocardial infarction and stroke in childhood. And these are the most accelerated forms, of course. And then prevent the early death and the burden of quality of life. The clinic that we have at MGH, with Dr. Peer, Dr. Al-Hadid, Brian Wisher, Grabowski, Nader, and now our new MP, Erica, here, is seeing kids that have had strokes at birth. before birth, after birth, at 2 years of age, at 5 years of age. You can imagine and picture what is developing with brain injury, plus all the disability that can come from a lifespan. Not a person at 75 years of age having a stroke with whatever is the lifespan thereafter. So can we group them some way? Because it kind of becomes overwhelming. There is 55 monogenic vascular disorders. Where do I put them? Yes, we can. We can do it by phenotype. Cerebrovascular disease, if you want to arrange a few of them. Cerebral vasculopathies, I gave you a couple of genes. You're familiar with moya-moya, RNF231. NF1, MYH11, milk, some HDA do cause the same disease, ACTA2, steno-occlusive disease of the brain. So you see same biology, smooth muscle cell proliferation, intimate hyperplasia. COL4A1-2 can produce vessel wall dilation, vasculopathy. They also can cause a small vessel disease, you see in the third line, together with other genes that we know alter endothelial and barrier function and can produce small vessel disease in the brain. And of course, the vascular malformations. And you have the HHT genes here, all the cavernous malformations and AVMs, even sporadic AVMs is caused 60 to 80% of the time by a single somatic mutation in KRAS in the endothelium. So that is something to be also ponder, how much of our diseases that we call common or non-genetic are not in reality the somatic mosaicism that is really causing, in particular, these kind of malformations, tumors in the disease itself. And then systemic vasculopathies, and you get aortopathies. So you can think about those that cause aortic dilatation, thoracic familiar aneurysm, a lot of connective tissue disorders. The same genes repeat. I don't know if you see it. We talk about the continuum. The phenotype might be different because the vessel anatomy in the wall is different. So there is a different biology that is affected by the same gene and the different pathways. And then again, the same vascular malformations that we talk, and I'm most concerned about the brain because of the disability, occur in the body, and they also can be disabling. So this is a complex slide, but it's trying to summarize. And from here on, I'm going to be very focused on one disease. So I promise to stop talking about general terms. So you can see what we really are doing. So we need vectors. Vectors we call what can get us there. Okay, so a nanoparticle or an AAV. This is our picture for AAV from now on. This is our picture for nanoparticle. We want to do a customized gene therapy for the need that is targeting the vessels themselves. And these are the mechanisms of dysfunction that we're trying to tackle. All vascular disease in humans is caused by these three mechanisms: smooth muscle cell dysfunction, calcification, and angiogenesis. And here's where also endothelial dysfunction becomes a problem. So we pick a prototypic severe form, and we didn't pick, they picked us, by the way. I'm going to be very honest. These are the patients that have hammered us from years and years of, can you help us? Can you develop something? Can you do something for our disease? And one of them is generalized arterial calcification of infancy, prototypical atherosclerotic calcific disease of infants, 60% mortality in babies at 6 months. Smooth muscle cell dysfunction, the prototypical most severe form is a mutation in the actin gene for the smooth muscle cells, that's ACTA2. And I'll tell you, that's the one that we're going to focus and we're going to go after with a very elegant correction and see if we can learn something from that. And then the brain AVMs is our initial indication for things that involve endothelial dysfunction and angiogenesis pathways. If you haven't done genome editing, these slides are from Ben Clanstiver. Thanks goodness, he can talk. And if you can see him, he's at the Center for Genomic Medicine. He's part of the Department of Pathology at MGH. And the enzymes that you hear, Beam, Verve, Intelia are putting into humans are his engineered enzymes with the base editors that David Liu has developed in combination. And ideally, and this is what everybody wants to be doing with precise medicine, is you identify what the problem is in the genome, you correct the problem, and most of what has been done for treatment right now and is widely enlarging is ex vivo genome editing correction. If you may be familiar with CAR-T, that's how we use genome enzymes, genome editors to correct the cells. But what we are tasked to do here, because we cannot take the vessels outside, and we also try with bone marrow, and I have shown also that they might get to the brain, and bone marrow progenitors may contribute to some cells in the brain vasculature, but not enough to correct the disease. So we got to go in vivo. That's what that needle means in the arm. It's not a vaccine. It's a needle in a vein. We need to go systemic delivery, and we have to be able to use these enzymes. The repertoire of tools is enlarging. As I'm speaking, there is under review current papers in Nature and Science that we are part of or know of that are going to change even what I'm showing you in this slide in the next 12 months. It takes 12 months to publish those papers, but they might be in bioarchives in a couple of weeks. You can edit DNA. So you have a base change, missense mutations, most common type of mutations in human disorders, 7,000 rare diseases in the human, more than 400,000 missense mutations. So if you get this to be scalable, great. We can do one at a time, elegant, one single change. I don't cut your DNA. I don't use repair mechanisms. I don't exhaust the cell. That's the ideal. And the efficiency of this little thing tagged to the CRISPR enzyme, which is the deaminase, is the highest for all of these enzymes. So highly efficient, very specific and targeted therapies. If you need to correct more than one letter, you can do prime editing. It's a small segment that you can insert in that or replace with a guide into the DNA in that specific place where you want it. This is not the same of lentiviral gene addition, where the gene inserts anywhere in the genome and is totally out of control and can cause oncogenicity, which is what we are seeing with a lot of the gene therapies, including the one that we have approved for cerebral ALD. We have the risk of integration in random places that can cause oncogenicity. This is different. This is where the gene has the thing, is under regulation, and it only can be done there because the guide is driving the enzyme to that site. And I'll tell you a lot about off-targets. This is not an easy thing to translate. We can insert larger sequences of DNA, or the challenge here is immunogenicity to DNA. That problem has been solved. You're going to see it published in the next few months, okay? You can deliver DNA without innate immunogenicity in vivo, and that's only in mouse. So again, nothing here has crossed the line to humans, so join me in the quest, and let's not sleep many times after we dose the first patients. It's going to be hard. But we have to do this work. The tools are ready, and the horizon of doing a lot more precise and less and safer therapies is really there. And then you can also regulate. You use the enzyme to recognize a particular promoter in cancer, and you can methylate them. You can sit on them for a particular amount of time. This is already in use in the agricultural world, largely. In the laboratory diagnostics, largely. This is not future. This is current past, okay? So this has been in use. It's not been used in humans. So our criteria was first in human clinical trial criteria. If you don't meet that, we cannot work on your disease yet, because we want to take things to clinic. We will put work on the disease to characterize it to get you to the human clinical trial criteria. So we need a rapidly progressive, severe disease. Not too severe that we cannot tolerate the infusion of a genetic therapy, okay? That's a little caveat there. Known markers or biomarkers of disease progression. A lot of what we're gonna be doing is short-term. We're not gonna get to a stroke as an outcome, and I'm gonna be watching for that in the next decade, what happens or doesn't. But you cannot use that as a clinical trial endpoint, even when it's the most desirable one, because diseases don't behave that way most of the time. Hemodynamically stable patients, that's the point of like a certain bar needs to be met. And then we conceptualize what could be the solution from the genome editing side, and this happens in silico. And this takes, in Ben's hands, for all of these, maybe a couple of hours. And he even has now an AI way to have the enzymes be further mutated to really target the specific mutation and decrease the off-target effects. So, and he's not the only one. So this is coming. We are getting to the point where you can model, if you can tackle that specific change in the DNA with a precise way, with a minimal of targets. This is the real challenge. We haven't yet shown that we can get to these smooth muscle cells in the arteries, the veins, and as you get into the brain, perforating artery here, branching into different arterioles, the capillaries, changing the blood-brain barrier. Even better, one of the things that I wish to see in my tenure, using the vasculature in the brain, Vern Caveness used to say, 1 capillary every 8 micrometers, and that was Padilla's work, and you can have the book if you need it. Which means if you want to deliver something that is secreted into the brain, and you can get it to be produced by the vasculature without damage to the vasculature, you have the best coverage of any delivery route that you could ever dream. And you can also think about the same is happening when you cross in the blood-brain barrier, when they're using the transferrin receptors for delivering oligonucleotides or delivering AAVs, you're just using this widespread coverage to really biodistribute across your brain. So both ways to use it to deliver something or to use it to produce and then deliver something is a horizon that is achievable. There is mouse models that have been corrected with lysosomal storage diseases. So this is not science fiction. This is really proof of concept in vivo has been done. I can tell you we're very excited. The nanoparticle has now moved into indications in the mouse, meaning we have a nanoparticle that can deliver both DNA, they're polymeric and lipid doing the job, DNA and mRNA to the smooth muscle cells and endothelium in vivo in the mouse. The brain side, they don't cross the blood-brain barrier. So we're getting really control of that other side effect that we don't want to be messing up with neurons and astrocytes with long-term changes that may not be needed in those cell types. So we're seeing what is happening. There's a lot of these nanoparticles in the liver. We have to detarget the liver. If you're not interested in the liver in your disease, which we are mostly not, okay, then you may want to decrease the transgene expression. And this is where we are with the nanoparticles. There's a lot of AAVs out there, and you probably have heard SMA, which is the most. There are more than 6,000 babies treated now with gene therapy, adding the gene to motor neurons and other things. And as you know, there is other AAVs at different stages of development, targeting different parts, but there is no AAV that can target vessels reliably and pericytes in the brain. Casey Maguire was trying to develop an AAV that could target microglia, and in the process, he stumbled with a picture that looked a lot like this, and is probably, you can see in the white, this is a brain coronal section of a mouse that has a lot of transduction of vessels and almost not a single neuron. There's a few interneurons here and there that are getting transduced, but a very specific targeting of endothelial and pericytes and smooth muscle cells. And when I saw those pictures, I said, can I work with this? He helped us develop it further. We can now bring it into human arteries to do some of the very basic mechanistic stuff. It's like, does it transduce these cells in the human? Does it transduce it when you're in perfusion inside a vessel? You have to cross endothelium. Does it even cross endothelium? And the answer is yes. We get 100% coverage of smooth muscle cells with very low dose ex vivo. Again, that's a different bar than putting something in vivo. And one of the most difficult organs in the vasculature to tackle is the aorta. Every cell of a smooth muscle has a layer of elastin next to it. So elastin is like a scaffold, and in between is the smooth muscle cells. Just imagine you trying to get through to cover 300 cell layer. So we were very happy to see in mouse, which only has 12, not 300, that this vector could really cover most of the ascending aorta, which is where most of the disease occurs in humans. And that was having a transgene expression that was very robust. And this is a CRISPR editing strategy, is a Cre enzyme delivered with the AAV that then acts on a TD tomato reporter mouse with green fluorescence. Jude Samolsky, again, the VILA goes way beyond NGB. The VILA goes to people that really want to help you bring these therapies for these kids. Has taken us on, did an in silico. Jude Samolsky is the godfather of AAV. If you guys haven't met him, he gave a talk, the keynote speaker at GCTI last year. Hopefully that's recorded because it really shows the steps back that occur when you're starting to dose humans with AAV and how you learn your lessons that only can be learned from dosing humans. He took our AAV PR, and because it's derived from AAV9, compare it in what it would look like to manufacture at large scale, and the answer is awesome. It has the same profile, thermostability that the AAV9 has. And AAV9 is the most known viral vector that we know how to manufacture. That's why it's important. What about immunogenicity to the AAV? Can we predict that? There is a way with the epitopes that they will react to. And you can see here, they're almost identical between AAV-PR and AAV9. So prediction, this is in silico. And you can see from AAV9, which is the yellow part, AAV-PR only has this peptide that is different. And we got really lucky because it has super high affinity, and Sue has been doing the mechanistic work in the lab to show that really is higher binding to one of the principal AAV receptors, one. Two, it has a homology with a protein in the cytoskeleton of mesenchymal cells, and that's where we get the specificity. And this is what it translates to, and we are going from 7 to 15-fold higher efficiency in vascular and smooth muscle cells human, pericytes human, and the arteries that I was telling you about in the bioreactor. So arteries from patients coming fresh from the OR, ABPR outpaces the transgene expression in the to AAV9 in those. So taking a specific indication and going through the whole arc of trying to develop this all the way to the clinic. So if you think about smooth muscle cell vasculopathies, and I call them this because I need to settle on what cell I need to target. That's how I categorize things. Either the pathway, the gene, or the cell that needs to go. But in terms of phenotype, we know that these alter white matter. They usually affect pericytes, the cousins of these smooth muscle cells, and the blood-brain barrier. So you get a small vessel disease. You also get a small vessel disease because your arteries have lack of autoregulation. The smooth muscle is not contracting and relaxing the way it should. There is loss of cerebral autoregulation and neurovascular coupling. And in the large vessels, unfortunately, smooth muscle cell, most of the time when it is functional in a muscular artery, will proliferate, meaning will narrow the lumen because it will produce intima hyperplasia. And that is what leads to the arterial ischemic strokes that are devastating in these disorders. And of course, you get, I call it vascular dementia here, but these are 12-year-old kids. Okay? So I cannot call dementia. And when I put the grant up, the first thing they said, doesn't live long enough to develop dementia. You cannot call this dementia. I would love the criticism. I was like, okay, cognitive decline. Let's call it something you like. I understand that we need to let the mouse live longer if we want, but our mouse is very severely affected. So we took the approach of going with a base editor. There is a single recurrent mutation, arginine 179H, that you can target by a deaminaser. This is an angiogram, and I'll tell you more about the disease phenotype in the patient. We need to, it's an autosomal dominant, it's a dominant negative effect. You cannot do okay with the dominant effect of the mutant protein. So you gotta get rid of the mutant protein. The one allele that is healthy is not sufficient if the mutant protein is around. You could produce a knockout of that allele. That was one possibility. We take it also as a backfold strategy, but it's a little bit dirtier because it cuts DNA. It has a lot of more off targets. Base editing, much more precise. So this disease does not only affect the brain. This is what an angiogram as coming out of the heart, the valve, aorta, ascending, arch, descending, and here are the iliacs. Your vessels you know the most. These are the common carotids. This is what it should look like. This is an MRA. I changed you the technique, but you can tell that this is fusiform, dilatation, aneurysm of everything. Subclavia and carotids. And if you notice that these carotids then narrow and disappear in the brain, and I'll show you now the brain. But let me put it in the way that I keep my energy to be up all day running, which is my patients. This is Rini. She came to us after all her strokes have occurred. She's a patient from New York. And this is her. We didn't need the sound. She's singing Frozen. And she's around 8 years of age, 8.5 years of age. She did not know the disease was in her, even though she had a large PDA repair with a sternotomy at birth, plus fixed dilated pupils. And I just gave you how you diagnose these patients. Fixed dilated pupils and a PDA. Please send the gene. There is no differential diagnosis. There are only milk, ACTA2, like 3 genes that can do this. So we know that these patients are born and the vasculature more or less looks like this. This is at 10 days of life. You get the carotids. That's not too abnormal compared to a control. It's not that dilated. There is no siphon for the carotid, and there is straightened vessels for sure. You can recognize this pattern, but there is no narrowing that is critical. We did a retrospective with Arne Lauer and Diana Milowicz, a study to see what was the effect of time over this vasculopathy in children, 27 children across the world of a disease that only has 55. And we found that there is a ratio in each patient, which is this one here, between the petrous portion of the ICA and the terminal ICA. When that is more than 70% stenosis, we see large arterial ischemic strokes. When that is not 70% relative stenosis, we do not see arterial ischemic strokes. We do see white matter disease in all of them. Unfortunately, she went on to have an EDAS because she was having symptoms of TIA, and she developed back-to-back strokes, and I will come back to what her strokes look like in an image in the next couple of slides. But this is Rini, a few months, this is more 18 months later. And then this is cortically blind, unable to speak, unable to stand, only using the left hand. At the moment, she's 19, bedridden, ventilator dependent, still having a lot of meaningful life, but every day is a given because she has dissected her carotid in the neck intracranially, has more than 10 strokes since the first one, and we can't stop the disease whatsoever. This is in a nutshell what the disease looks like and how we're thinking about our gene repair or gene targeted repair. So this is the mortality. By 14 years of age, half of the patients have died with this disease that we know of. And then we know that the risk of stroke is higher in the first decade of life. Those are where the events are happening. And the aortic dissecations are happening as the aorta dilates over time in the second decade of life. So I just gave you what are the big two phenotypes that we want to modify. And then we found this severe ICA stenosis as a marker that occurs at 2 to 3 years of age. They're not born that way. They develop it over time. That's the intima hyperplasia of the smooth muscle cell dysfunction that I was talking about. So we really believe there is a true biological window for correcting this smooth muscle to prevent it from reacting the way that reacts to shear stress. If we can get the aorta, it will also help so you don't have the dynamics of flow. There were 33 kids with a disease known, published in the paper of Regalado before Rini. There are 31 and counting, and the last couple of weeks, we are hearing about 2 to 3 almost per week that are being found through the connection with the foundation. They don't come to me in clinic. They don't ask for an appointment. It's a parent that is saying, we just heard about this patient. Let us connect you with one of the groups that has expertise. And it's a lot more easy to diagnose when you have papers out there completing our academic role of publishing that show what you need to be looking for in these patients. So I told you they have fixed dilated pupils. This is what they look like. Fixed dilated, 8 millimeter, no questions asked. It's not the same in a newborn. It's not easy to see the pupils in a baby that is just born. But if we can add to newborn screening, the exam of, you see, red reflex is part of every discharged baby from a hospital. If we could say, do pupils react? Check. We can diagnose these patients right there. And the best part in our research, and Claire Fong and Savia have taken this to the next level, is that there is a retinal vasculopathy. There is tortuosity. There is increased peripheral perfusion, like kind of what happens in retinopathy of prematurity. This will be graded a grade 5. If this was a newborn, this is a 7-year-old. And I put a control at the bottom just to show you what the arteries should look like more than what they look. I told you about this ratio. This is the 70% relative stenosis and the arterial ischemic strokes. This is a control cohort. So if you have any doubts that this is a mechanism that can happen in humans, no, that doesn't happen. Petrous arteries in ICA petrous does not dilate. You may want to say atherosclerotic disease or anything can change the diameter of your terminal ICA, but the truth is this is a specific ratio, individual, you don't need a control, that if it changes over time, you know that you're approaching the high risk. These are the patients. So we organized these meetings here in Boston, 2019 and 23, and literally we've been working on what to target in their quality of life. This is work that has been led by several people, CRCs, that Annalise is trying to wrap up. And the one thing that I wanted to point out is the mean time of diagnosis was 11 years of age before we existed as a clinic and all these papers started coming out. And now we lower it to median of 3 years of age. So it's happening. And most of the patients we're seeing now are babies. So we are getting to modify care, which is going to change our design in the trial over time. And something that is completely missed because they're so sick from other things is that they do have a lot of developmental challenges. And this is going to be the first time. And if you ask the patients what affects most the quality of life is the neurological changes, either from stroke or the developmental milestones. And then the next one is the cardiovascular. They have these open heart surgeries to fix the PDA, fix the aorta, get the replacements. We just discharged a patient 2 days ago from our wards that have another aortic ascending repair. So this is what we want to be doing. And we have to put this enzyme into 2 AAVs because it's too big to put it in one. Before we did that, we created a mouse model with the exact mutation that we needed to recreate in the disease. This is a smooth muscle cell high resolution cytoskeletal picture. You can see actin at play is the green stuff, and it's polymerized. So it becomes part of the F fibers, the stress fibers in the cytoskeleton. You can see what happens with the mutant present. it does not polymerize. So even though you have healthy protein, you cannot do cytoskeletal action, you cannot contract. Transcriptional dysregulation leads to proliferation and secretion of collagen. That whole biology, Mark Lindsay has studied for decades and has published specifically for this mutation. Got lucky. It didn't get lucky with ALD on creating a mouse model and spend a lot of time. We did get lucky on this disease. We created a knock-in, and we have the terminal ICA stenosis. We have the dilation of the aorta. We have the abnormal. These are now the cells from the mouse. They look exactly like the ones from the human. And this mouse is dying very soon. It's mostly the gut. The gut is a smooth muscle and doesn't move. So they get impactions and sepsis and they die. So we needed to fix the gut if we wanted to see survival. So we were hoping that we get there. And then Mark does these fancy echoes to measure the diameters of the root, ascending, and carotid arteries in the mouse live without killing them over time. And David and Taka have taken us on really characterizing the cerebrovascular anatomy. This is the narrowing of the ICA, MCA. It's exactly the same thing. Remember the straightening of the vessels in the angiogram that I was referring to? This is a control. This is an ACTA 2. The mouse has the same thing, premature bifurcation, straightening narrow vessels. We do MRI, you see the same thing. This is the ACA, this is the MCA, this is being quantified, and is work happening right now with collaboration with Jean Ju Lab at Charlestown, and Lydia and Joanna have taken the lead with David supervision. At the histological level, this is what an artery, when it has smooth muscle cell function and contraction, should look like, wrinkled. This is what an artery on a patient with ACTA 2 is an MCA looks like. And you see the proliferation of the intima. There is no wrinkle because there is no contractility. And this is the staining of that in the carotids and in the aorta of that patient. And you can see what happens in our mouse model. And it's pretty much the same. Small vessel and large vessel has the same pathophysiology, shows the same phenotype. And we don't see the wrinkle of the arteries because there is no smooth muscle cell contractility. I show you what their cytoskeleton looks like. The white matter disease is in 100% of the patients present. It does not progress over time. That's why we are not using it as an endpoint. And in the mouse, we see severe loss with leukorrheosis and pronounced perivascular spaces, which we also see in the human. We quantify a lot of that thanks to all the help from Vijay that has been leading many of this. We know the blood-brain barrier is disrupted by perfusion imaging on the patients. We do this in the mouse. We find the same thing. We have increased blood-brain barrier permeability, parasite dysfunction, and we have also microglia activation and astrocytic response, mostly perivascular with astrogliosis. And then it comes the very fancy part, which is necessary. So we can say that functionally, we have corrected this mouse and are doing something that is relevant to the physiology of the brain. And these are optical imaging and different technologies that Cheng Ayata and David Chang have been developing for years in their lab. And it allows us to assess cerebrovascular autoregulation and neurovascular coupling live without killing the mouse. Remember the patient I show you? This is her little TIAs here. This is the first stroke. They decide to do EDAS with the best neurosurgeon we know in vascular, so everything that could have been done. And then not right after surgery, 30 hours later, she starts doing ACA, bilateral ACAs, and you can see how this spreads to the next area. If you are a stroke neurologist, this looks like they're moving to the penumbra. The next area in DWI over time is the next penumbra. That's true failure of cerebrovascular autoregulation. And you can see the vasculopathy is not mild, it's very severe. We do this in the mouse with bleeding, complete failure of cerebrovascular autoregulation. And then this can be done even transiently with ligation of the carotid. So you can see how the drop on the cerebral blood flow occurs. And that drop is much higher in the ACTA 2 mouse, and it also does not recover right away because the circle of Willis, which should be able to compensate quickly with vasodilation, vasoconstriction, cerebral autoregulation, is failing. There is no smooth muscle cell function. These things are critical. And what I love about this, there are careers here to be continued. So it's not about just getting to the clinic and doing a treatment. Doing the base editing is an art and a work that requires an engineer. And this is what you want. You want high editing on target, that's your mutation, with very little off target, that's the bystander. So the editors have a window and they can edit more than what you want. We want to edit this A, not that A, not any of the other A's in the genome. So that's what is represented here. That's the on target, that's the off target bystander. So we play with different enzymes, we play with different guides. I'm talking about dozens of combinations, and you kind of like want to walk away. This happens in 2 weeks. Okay. So if you have a reported line, you can do really quickly these assessments. So we're accelerating these timelines. It's Ben's like idea of translation needs to be scalable, and there's a lot of things that he's doing to move this along. Oh, but it's too big. It doesn't fit in the AAV. We need to put an intane approach. So we split the enzyme, put the guide, and then we show it works the same way in vitro. Let's see what happens in vivo. We don't trust our novel AAV. We do know AAV9 is in clinic. So if we can do something with AAV9, we'll move with AAV9 to clinic. So that's me trying to treat the patients, doubling the cost of everything, because we need to do both. And this is what happens. AVPR is more efficient in editing the liver, even that. And of course, the brain vasculature, and here is the heart. And then we have data coming up on single-cell RNA-seq on the aorta to really understand what's happening at the vascular level. It rescues the neurodegeneration. There is no white matter loss in the mice that are treated. And we're treating them early. We treat them at P3, neonatal. You treat them at P14, you also see rescue, but we cannot let them live long enough sometimes. So it's not as efficient as a P3 treatment, as we also predicted. I told you that there was this blood-brain barrier disruption, astrogliosis. I'm showing you the hippocampus. There's severe neuronal loss, by the way, in this disease. And all of that is restored after treatment with AAVP9 or AVPR, delivering this base editing. And this is a little like a snapshot of microglia. This is what I was mentioning. P14 can also improve, but it's not as robust. These are the short-term studies, but these mice here are 3 months of age. And you can see the mutant labeled there in ACTA2, small, very small, failure to thrive, cannot move, cannot do rod. We have all behavioral batteries on these mice, and this is the littermate treated, and you cannot tell them apart. And now we have this long-term, high dose, low dose, so we are learning a lot of things. We are able to extend the lifespan more than 5-fold, and we even got to treat the gut. We didn't expect it. This was the most surprising thing. I was not expecting the pupils of the mouse, which are fixed dilated at birth. Beautiful that it recapitulates even that, to have a more normalized pupillary function. I did not expect the vector to get to the iris. I have no idea. I know nothing about the eye. That's when we enroll a lot of experts across the world to help us. One in Australia, one in the UK, one here, to see how we can move this to the next station, because that could be our biomarker, immediate biomarker of effect, if we really have a therapeutics. And the arteries in the retina are the brain arteries. So if you think about it that way. Toxicity and the effects in other organs, we even fix some of the hydronephrosis that is caused because of the hypotonic bladder, that's also smooth muscle. And it's not causing severe liver or any toxicity that we can observe in the mouse. The off-target effects is 20 times more labor than finding the enzyme that does the job, just to say that. And you even have to recreate an assay where you can see what does it mean to change that particular amino acid in the protein to the cell of interest. And those are very careful things. We had to put this in front of the FDA when we do a pre-IND. We did already an interact. The pre-IND is coming out in the next couple of weeks. And in summary, we've been able to restore the vascular anatomy and the function of the vessels, diminish the neurodegeneration. We even fixed the aorta. I saved you from that just because we are in the neurology grand rounds. And then we are trying to go from mouse to babies. So we're trying to really leverage everything we can to de-risk these therapies and understand what we need to be doing. 2 words on a gene addition. This is simple, 2 slides. This is the one that I told you is atherosclerotic calcific disease. It's a loss of EMPP1 function, which produces pyrophosphate, which inhibits the calcium accumulation. 6-month-old, and all the vessels are calcified. This is a CT without contrast, and you can see every vessel in the body pretty much. And they narrow and they cause MI, so myocardial infarction. We developed several strategies. Do we do a soluble? Do we do a transmembrane? Do we deliver to the liver and then we use the liver as a machine? Are we able to do this? The answer is yes. Now we have proof of concept. We're moving this also to an interaction with the FDA. And we've been able to prevent the calcification and the demise of these mice all the way now up to around 9 months of age. So coming, enzymatic activity is restored, and the biomarkers in plasma are coming. And why is this relevant? Because it's one of the EMPP1 and its pathway are 4 of the 5 genes that have been associated with calcific coronary artery disease, and the largest GWAS cohort in humans. Loss of function of these enzymes cause acceleration of that calcific atherosclerotic disease. So this is a therapeutic that could move to a non-genetic disorder directly because it's part of the pathophysiology. There is a VILA. I need to update this slide too. This is my lab and Mark Lindsay's lab. We ended up working in a synergy. That's how we do it in the clinics. That's how we're doing it now at the bench. We're doing everything at the same time of doing natural history studies. This is where all becomes crazy because you need a real clinical team to be able to deploy natural history, clinical trial readiness to be able to treat these patients. And these are leading clinical trials. So these patients are being followed and their assessments are used to measure what we're targeting for the therapeutic effect. So they're very important. We're doing the same thing with ML4 at the moment, and we are using every piece of data that we have in our natural history study. This is very complicated. I wish I can simplify it. This is a multisystemic disease. For most of the other ones, I can narrow this to 3 tests, okay? All the other vascular genetic disorders. And there is a lot of research done ongoing to what do we do now with these patients on the clinical management? This is revascularization surgery, and we do not see a lot of angiogenesis. And the worst part, we continue to see strokes and white matter injury despite the surgery. So there is an ongoing research effort internationally that Diana and Emily and a parent of a patient, Angela Cass, is leading on getting all the surgeons that have done surgery, plus all the parents that have, and the patients that have received it to see what is happening. When you try to get something to clinic, it's really more than what you do in your bench. Be aware of this and not just be aware. If you want to do therapeutics, work with regulatory people. I tell you because you change the design of your experiment at the bench and what kind of things you need to be doing now, because by the time that you're ready in 2 years, if you don't have them, you do not have them, you cannot do anything with the FDA. So you had 2 years behind. So and then again, this is the dream. We won't realize any of this without robust funding, someone else taking it on and making probably a biotech, and also demonstrating safety first in humans and the ability to target the vessels. And we have pyramids for everything, for every single gene. And I can tell you how we grow the programs to more common diseases and how many more applications. But to be honest, everybody wants to see human data before they dive into something like this. This is my thank you. Before last slide. So a lot of people, most of them started working when we were postdocs in someone else's lab. So never underestimate your networking potential as a resident, as a postdoc. And that is the relationships that really make people vibrate, learn to work with each other, and then explode from there. And this is not possible without an institution that has the know-how, the resources, and the willingness to take crazy ideas forward. More importantly, the Department of Neurology supporting crazy faculty. Like, we really are good at hiring people that are not doing what no one else is doing. Okay, so you are already taking risk. This is taking even more risk, given your resources that are not many. And here we have our team, and you wonder, what is this Angea Biotherapeutics? That's Charles Roswatt. He's basically an angel investor that decided to do this way to be able to get the IP, to be able to be put in front of people that can give us the next round of funding. And it was a blessing. It pretty much matches what we have from NIH in funding over the last 3 years, and he continues to support us to these days. And this is one of the pictures at the first year report on the 3-year collaboration. I did not mention that we clicked gear on this in April 25th, 2022. This is a 2-year live program. Yeah, that I had the same. And the mouse model was done, arrived to the lab December 2021. Like, that's when we got the mouse model. So it can be done. This is Bernard Kinan, one of our pulmonologists. Never ever give up on kids. And I learned my lesson with these patients. I'm a critical care person, and man, if your PaO2 is 35, 40 for 5 days, your brain needs to be cooked, no? Like, when you wake up from that, it's almost like anoxia for 5 days. These babies, these kids are hypoxic to that level, to the 40s for several days. We're resuscitating them, and they wake up neurologically the same. Okay, the white matter is getting hit. Maybe we may be losing neurons in the future, but I cannot tell you how much I have learned to humbled about what I know when I'm trying to manage these patients here at MGH or elsewhere in the world, because they crash anywhere and they call us, and our PQ team picks up the call with me, with Mark, with Bernard, anywhere, anytime, 2 a.m. in the morning, kid having surgery in France, to brainstorm with the primary team that is trying to resuscitate a kid that is not going in the right direction. So with that, I hope to take a few questions. Thank you.
Betty, that was a great, inspiring whirlwind tour of a lot of information. What is the reversibility of the ACTA2? I mean, you showed the cross section of the vessels, but did you get like, for instance, your 70%, critical ratio? Does that change in the mouse?
We don't see stenosis. We haven't been able to quantify it. We're running short of numbers of mice. So there's work ongoing on the different parts. Not every mouse serves every experiment. So we haven't gotten the quantification, but we see in an MRI preliminary data and an Indian ink that these arteries are like normalized.
And do you think you're normalizing gut function? Is that why the survival is improving, or?
Yeah, so they don't die from stroke. So the gut function, I do have a little slide here. Yeah, let's not do it. So the gut function is working. So from no motility and large fecal, cecum, colic impaction. In fact, Alan Goldstein has taken us on and is unraveling the innervation and what's happening. And we see now with the treatment that they have peristalsis, and that's why they're not getting impacted. By 9 months, either they get a UTI or they start looking, they stand it, and the animal facility asks us to kill the mouse, to euthanize the mouse. So we haven't seen death from stroke up to 9 months.
Great. So what about, so the gene screening or newborn screening for something that's this rare, so 55 in the world. I mean, I don't know what the true incidence is, but so pupillary stuff is just going to create like hundreds of thousands of false positives probably, right? I mean, for every real thing, you're going to have to do this as genetic sequencing, it seems.
Yeah, and I would not do anything. So this is just the same thing that happens with the red reflex that is not there. They get referred to ophthalmology. There is no sequencing. There is no genetic consult. Like in the exam of the newborn before discharge, we do red reflex for cataracts, congenital cataracts. And then you have to refer them, and it's just, so you just have to add that. And then that person has to determine this is congenital mydriasis. And if that's the case, then the genetic testing gets sent from there. And that a few patients have been diagnosed by ophthalmologists. That's saying like, hey, these people are, and the kid already had an aortic disc replacement, a PDA repair, they have got this motility, Moya disease, and nobody has thought about
I get all that. Just in terms of screening for something that's that rare, the false positive rate, even, with good people, is going to be super high. And it just seems like going directly to gene sequencing is your best chance.
There is a project in California funded by the Ritter Foundation, the Aortic Familial Thoracic Aneurysm Foundation, to do genome sequencing, whole exome sequencing on newborns in California for 1 year to see the true incidence of all vasculopathies, not just like ACTA2. I didn't mention, but there are more than 2,000 patients per year with ACTA2 mutations with thoracic aortic aneurysms. There are other 15 mutations in the gene that can give you the aortic disease. By 2030, you dissect. We have several of those patients. We are main hospital for aortopathies, genetic because of Mark. And those are much more frequent, but they don't have the severe pupillary distension. They don't necessarily have a large PDA at birth. So those get more challenging even for diagnosis. You just get diagnosed either symptomatic or by family history. But it's autosomal dominant. So that's why there are 2,000 patients per year in the US, because there are families that have this disease. So this could open treating these severe, very few patients. I'll tell you the counterpart of treating these patients. First of all, they need something. This is horrendous, like many of the diseases we all deal with in the clinic, for these parents to put these kids to bed and have to wake up to a stroke and things like that every day. So there is a massive unmet need and a lot of things that we can do together. And we even have a small molecule going on. We also been doing in parallel small molecule and siRNA, because the siRNA will cross the gene. It doesn't matter if it's ACTA2, NYH11, TGF-beta receptor. it would just normalize smooth muscle cell function at a much downstream, that transcriptional downstream that allows, is being shown in transgenic mice, but it hasn't been done therapeutically, to cross monogenic and non-genetic and many of the monogenic. So in a way, we're using it as a prototype of severe form of a disease. And if we can get better on this one, meaning make patients live longer, modify vascular biology, we now have the tools to really move it into more common mutations or the other mutations in ACTA2. Every mutation is a drug. So that's what we're trying to also work with the FDA. Can this work be done in a different way? If you're treating the same disease, same phenotype, can I alter the guide and the enzyme, show you all the off-targeting human cells? You have to show me where else you're editing in the genome, not just that gene. But if you have safety, can we do it without having to do the animal model? And all these are very lengthy, expensive. We got lucky. This doesn't happen. Like, we've been working with ALD for 4 decades on the mouse that we cannot get it to develop cerebral inflammation. So we did everything without a mouse model. So ideally, we move the field by trying to tackle these patients. I am not proposing whole genome editing. Might be coming from other sides, but not from our vasculopathies. I'm aware of efforts in getting newborn whole exome, but. Right.
Great, thanks.
Thank you.